Hey, welcome back out to the channel. My name's Kim. I'm a licensed and nationally certified East Asian medical provider and a Taoist practitioner. Today, we're going to talk about a little bit more about the science of turbocancers and could there be a link between the mRNA vaccines and turbocancers? Well, I don't know, but let's see what we can find out. First, I want to just kind of recap. Remember, this is a timeline that we're looking at, right? And this timeline starts way before the pandemic, gets to the pandemic, and then we get that letter from AstraZeneca right before the Pfizer vaccine is released to the public. We get AstraZeneca publishing this letter, and they voice a concern that maybe we don't really understand our immune system enough, and that right now the pharmaceutical companies are making their best guess at what an effective immune response would be with these vaccines, and they're basing that off of our history of vaccines. What's the problem with that? Well, the problem with that is our vaccine history is based on a completely different technology. It's like comparing riding a horse for transportation to driving an electric car. There's not a really good correlation there. Okay, so if we take that into consideration, let's do one more thing. Let's recap what the point of the FDA is, right? So the FDA is the Food and Drug Administration, and they're responsible, according to the FDA, for protecting the public. That's a big responsibility, and they're responsible for protecting the public health by assuring the safety, the efficacy, and the security of human and veterinarian drugs, biological products, medical devices, and our nation's food supply, cosmetics, and products that emit radiation. So that is what the FDA is supposed to do. They are supposed to protect our public health. Then let's get to it. Let's look at some of the Pfizer documents. Do you remember the Pfizer documents? It seems so long ago because so much has been happening, right? But it wasn't that long ago. It was 2020, 2021, the Pfizer documents. There was an organization of medical doctors and health professionals called the Public Health and Medical Professionals for Transparency. And they filed a Freedom of Information Act with the FDA and asked them to release the documents that were used by Pfizer for their emergency use authorization. There was a lot of stalling and a lot of dragging of their feet, and the FDA and Pfizer, they weren't really motivated to release these documents, and they wanted to take a few years because in a few years this would be a non-issue, right? I mean, isn't that how it usually goes? And so then these professionals took it to the courts, and the courts stated that they needed to release the documents immediately. And so that's what's happened. I think it was in February 2021 that maybe it was February 2022 that these documents were released. So we're 2 years into the pandemic. The vaccines were released in December of 2020. We've been uptaking vaccines for about a year now, and now they want to administer these vaccines to those 16 and under. I'm just going to go over a few of these research studies because every one of them is kind of consistent. It brings up some consistent questions for me and probably will for you too. So let's look at the first one. Let's start with the animal studies. And this is the R&D study report number R-20-0112. And what they titled this one is Characterizing the Immunophenotype in Spleen and Lymph Node of Mice Treated with SARS-CoV-2 Vaccine Candidates. So they want to know what the immune response is, what type of immune cells are responding when they get injected with these different vaccines. And they're also trying to identify which vaccine candidate they're going to use. So as we go through this, there's 3 key vaccine types. And the one that they're going to end up choosing is the BNT162B2. And that one, what's interesting about that is it's going to be the one that uses methyl pseudouridine in the mRNA. And we'll talk about that next, but let's look at what happened in this study. I'm going to blow this report up a little bit, so hopefully it's easier to read, but I want to go to page 27 because this explains what's being tested here. And we can see that the vaccine candidates are here. This is the one that we're going to use. And this one, this C group, I have never been able to find a definition of what that C group is, so I don't know. It never moved forward. What I was curious about is what's the control item? And the control item is going to be mice injected with sodium. There's 48 mice in this study. They're about 9 weeks old when the study starts, which means that mice live for 5 years. So it's about 3% of their lives. If we looked at 3% of a human life, that's maybe 2 to 3 years. And granted, okay, that's not really a correlation that you can make because these are mice and we're humans. But I think the thing is that these are really young mice and they're pretty healthy. So let's see here. Let's go to, we want to look at what's in the lymphocytes. This is in the blood, and it's going to show that there's elevated T cells in the blood. But I wanted to look at it in the lymph tissues, the spleen, the lymphs, the tonsils, what's going on there. And what they saw is that the response for the group that is going to move forward, which is going to be that BNT162B2, compared to the control group, it is significantly elevated T cell response. So this is CD8. This is CD4 T cells. This is the BNT162B2. Let's see here. When we're talking about T cells are those cells which are inside our cells to help protect our cells against viruses, bacteria, cancers, tumors. And then the B cells are what's floating around in the blood. The B cells are activated inside the lymph tissues. So inside the spleen, inside the tonsils, inside the lymph nodes. And these CD4 T cells help that happen. These TFH cells really help that happen. TFH cells really help the B cells differentiate into their antibodies. And you can see here that the response on the TFH cells is higher than any of the other candidates for this BN2-162B2. What they focused on was these TFH cells because those are so involved in differentiating B cells inside this lymph tissue, so helping B cells change into their antibodies. Moving down a page, now we see the B cell response. And here we see that for that BNT162B2, we have a really good B cell response, much greater than what the control group is. And I wanted to move over here to the class switch B cells. Out of the candidates, the BNT162b2, that has the highest number of class switch B cells. I am talking about this in Health Hub because there's so much science out there right now that's questioning whether this isn't a safety signal, that this wasn't a good thing, that maybe this was a safety signal. Let me talk to you a little bit about the germinal center of the B cells. But in the lymph tissues, when the immune system's activated, and remember we were talking about those TFH cells, those get activated, and what they do is they encourage this growth inside the lymph cells, inside our lymph tissues, the spleen, the tonsils, the lymph nodes, of this germinal center. So it's a germinating center. So they encourage this growth of this germinating center, and it's called a germinal center. What that center does is it allows B cells to differentiate. So that's one of the main ways that we get our B cell, our blood immunity, those things that are going to jump out of the cells into our blood. That's one of the main ways that happens. You can see for the candidate that did actually go into production that this B cell response is high. It's the highest out of all the candidates. The BNT162b2, really high here. It's very high in these class switched B cells. It's very high in creating those germinal center B cells. So it's very high in creating these centers to germinate these B cells that are going to create those antibodies that are supposed to attack the virus. So what do we know about this right now is we know that Pfizer selected a vaccine based on its immune response, and they looked for a vaccine that was delivering a strong immune response. But remember, in November of 2020, right before the vaccines are released to the public and after the FDA has given emergency use authorization to Pfizer, AstraZeneca comes out with this statement that says, hey, this is really complicated to figure out what a good immune response is because we don't really have any real good history. So whatever we're doing right now is our best guess at what would be a good response. It also appears that the FDA didn't ask Pfizer to verify or validate their assumption that a strong immune response was a good thing. It also doesn't appear that they asked Pfizer to verify if class switching, what was happening in that class switching, and if it was a positive class switch. What we know today is there is a lot of concern about IgG4 in the body. And if you are a member of Health Hub, I have an episode out there on the immune system betrayal. It talks about the science and what they're saying about this IgG4. And these IgG4, they are a subclass of B cells. And what they're finding is that these B cells are class switching from IgG2 or IgG3 or IgG1 to IgG4. And there's, in some individuals, a very strong IgG4 response. Now, there needs to be some more research done on it, but what it appears is that these IgG4 may be muffling our immune response to viruses, to bacteria, to cancers, and especially to COVID-19. I want to show you something. I want to just quickly look through one of the interim reports, and I want to look at the deaths. So they actually do this separate report on the deaths that happened during the study. And so let's just look at this really quickly because I think you'll see what had me confused, what I didn't understand. And page 3 is like the first death. And this person took the vaccine, had 2 doses. And they're a 56-year-old white person, obesity, hypothyroidism, depression, asthma, sleep apnea, acid reflux. They have a gastric sleeve, and they do have some tachycardia. August, September, October, about 2 months after the last dose, they die of cardiac arrest. And they say this isn't related to the study intervention. So here's the thing I wanted to point out to you. On the emissions, the potassium level's low at 2.8 moles. Now, there's another episode that I have out here on sepsis, because suddenly sepsis is like through the roof, the amount of sepsis that we have. And you'll see that the potassium levels are off and the magnesium levels are usually really off on this. Here's the part that had me really confused. It's unknown if an autopsy was performed. And what has me confused is that this is a study of a new drug, of a new technology, of something that we've never done before. Why wouldn't it be mandatory that autopsies be performed on all deaths? I don't know. Okay. How could you say that? So here's the investigator. In the opinion of the investigator, there's no reasonable possibility that the cardiac arrest was related to the study intervention, as death occurred 2 months after receiving dose 2. Pfizer concurs with the investigation's causality assessment. That sentence is going to be under every one of these. There's no reasonable possibility. And yet they didn't do an autopsy. So how could you have that kind of confidence? Okay, I'm going to go to another one. Here's another one, 2 doses. The last dose is in September. A month later, the subject goes to the emergency room. They also have a severe low potassium level, and they're showing heart failure, left ventricular heart failure. Now, we know that this is becoming more common today. There's no autopsy that's performed. Again, you see the 2 sentences. The investigator, no reasonable possibility that this could be from the study materials. Pfizer concurs. And yet there's no autopsy. How could you know? This person, let's see here. This was interesting because this one shows a reaction to the injection, and their last dose is in September, and they die of cardiovascular event in October or November 18th. So you can see the first injection, they got bruising. And the second injection, not only did they have pain on the injection site, they had fever. And here we go. No autopsy is performed. And the 2 sentences. There's just no way this could be associated with the vaccines. Now I'm going to jump down. You're going to see just on every one of these, there's no autopsy that's performed. And I just don't even understand that. How could you come up with this not being associated with the vaccines if you don't do all your science? But this one on page 48, this is an 82-year-old person, 2 doses. Last dose is on October 15th. I wanted to point out this one here, the vitamin D deficiency. If you've been reading any of the information on vitamin D deficiency, that's the one thing that I would suggest. Definitely take vitamin D. Even if you're not deficient, take vitamin D. Okay, this person dies of cancer that has metastasized. Weird, huh? Let's go here. So here's the thing. Their last dose is October 15th, 2020. On February 28th, 2021. So October, November, December, January, February. It's like 4 months later. This person's hospitalized because of pancreatic cancer that's metastasized to the lungs. They are in stage 4 cancer. And the study is not aware of any pre-existing neoplasm. You saw that their prehistory shows no cancer. There's no disease-specific treatment that's reported. The subject is discharged from the hospital on March 5th, and they die at home the next day. No autopsies performed. And again, in the opinion of the investigator, there's no reasonable possibility that this metastasized cancer could be associated with the mRNA shots. And yet, this is the exact thing that we're talking about when we're talking about turbo cancers. Turbo cancers come out of the blue. They're at stage 4 when they're identified, rapid progression, death. Okay. I wanted to look at one of the monthly summary reports, and this is the monthly summary report 4. It's for the month of March 2021. So let's just jump to page 34 and let's look at the summary data, because I'm going to look at graph B. And graph B is separated. The colors are separated by age categories, but graph B is about healthy individuals. This is individuals without comorbidities. If you look at this graph, you can see who the line that has the most issues here is that light blue line. And that light blue line is the 31 to 50-year-olds. So they're having significantly more events with these shots than anybody else. And who comes in a quick second? Well, it's going to be the 51 to, what is that? 51 to 64-year-olds. So there's this middle age category. They are getting a lot of events with these shots. That's going to be interesting as we look at these turbo cancers and who is getting the turbo cancers. Okay, I'm going to stop here because I have like 3 more reports that I really wanted to share with you and talk with you about, because for me, it showed some issues in the cells that I thought the FDA would have picked up on and said, hey, we should do longer term studies on this, especially since everybody in this study had this problem. But, they didn't. This is just going on for so long. If you want me to share more of these with you, put it down in the comments and I'll share other ones that I thought were just wild that the FDA didn't say, whoa, hold it, we've got to do some more study on this. And then I didn't think this was going to take as long as it did to just go over these 3 little studies. But the next thing that we'll do is we'll start looking at, after the Pfizer documents, what information do we have? What did we find out about the spike protein especially, and how that causes cancer, or how that might cause cancer. And then let's talk about that age range some more and who's getting sick. Okay, until next time, I'll catch you on the other side.